Hello there, my name's Daniel Mayer from the National Oceanography Centre in the UK, and I'd like to talk to you about some of our latest work that examines the question, can particle fragmentation by copepods explain observed particle flux attenuation in the mesopelagic? Before I begin, I'd very much like to acknowledge my co-authors, Wendy Gentleman from Dalhousie University in Canada and Tom Anderson, also from the National Oceanography Centre here in the UK. I'd also like to mention that if you are interested in this work, the underlying bioassays paper is free to download from the web. Please just Google particle fragmentation copepod and it should pop up. Failing that, please feel free to email me. So, why should we care about the mesopelagic? Well, I hope I'm preaching to the converted here, but very briefly, we know that the ocean's biological carbon pump helps regulate global climate by sequestering carbon in the deep ocean. And we also know that most of the particles of organic matter that sink out of the epipelagic are remineralized by biological activity within the mesopelagic zone, somewhere between about 100 and 1,000 meters below the ocean surface. Understanding this flux attenuation and how it responds to environmental change is really key to understanding future climates on Earth. Until fairly recently, attempts to quantitatively balance the sources and sinks for carbon within the mesopelagic had been unsuccessful. For example, as you can see in the figure on the left-hand side from Steinberg et al., the supply of organic matter, shown by the black bars, is well below the quantity of carbon required by bacteria and zooplankton, shown by the white and grey bars respectively. This frustratingly unsatisfactory situation was nicely summarised by the title of Bird et al.'s 2010 paper, What is Wrong with Present Calculations of Carbon Budgets. Thankfully, as you can see in the figure on the right-hand side, just a few years later, Gearing et al. Managed to balance the mesopelagic carbon budget for the long-term observatory site at the Porcupine Abyssal Plain in the Northeast Atlantic, at least to within observational uncertainties, which to this day remain rather large for the prokaryotes. This figure, also taken from Gearing et al., shows the results of our steady-state model analysis of the balanced budget. The most straightforward way to achieve mass balance produced quite a striking conclusion. As you can see highlighted in red, zooplankton intercept half of all sinking particulate organic carbon, or POC. However, almost 30% of the material encountered by these animals was subsequently fragmented and lost to the particle-associated prokaryotes. And this raised an important question. Why would mesozooplankton in the mesopelagic be such messy feeders in what we often think of as a very food-poor environment? Well, a possible answer lays in what happens to particles as they're fragmented. Back in 1970, Fentrell showed that benthic amphipods shred turtle grass fragments in order to stimulate the growth of the associated microbiome, which they then subsequently consume. You can see in the left-hand plot here that Fentrell's experiments nicely showed that the abundance of bacteria, flagellates and ciliates all increase as the size of particles decrease, owing to the increased surface area available for microbial colonization. And various other people have subsequently demonstrated similar activities in other benthic invertebrates, with Hilleberg in 1975 referring to this behaviour for the first time as microbial gardening. And from these studies, as shown in the cartoon on the right-hand side here, we hypothesised that mesopelagic copepods fragment sinking particles because it stimulates the production of microorganisms that have the enzymatic machinery to convert refractory detritus, a nutritionally poor substrate, into much more nutritious and harvestable biomass in the form of flagellates and ciliates. Coming back to more recent literature, earlier last year, Briggs et al. Used data from the Argo floats to very nicely show that particle fragmentation in the ocean plays a major role in regulating the biological sequestration of CO2 in the global ocean. And they stated that this may be the most important process in determining the depth at which fast-sinking organic carbon is remineralized. The plots here show the rates at which large particles are fragmented down through the mesopelagic, being derived from backscatter in brown on the left and fluorescence on the right in green. A particularly interesting result from this study was that approximately 50% of the observed decrease in particle flux with depth was related to the transformation of large, fast-sinking particles into small, slow-sinking fragments. And what struck me about this result is that our steady state analysis in the gearing paper indicated that zooplankton fragment approximately 30% of the particles that they encounter. And this got us thinking, maybe zooplankton really do have an important role to play in regulating the sinking flux. After all, a multitude of studies spanning several decades have already indicated that zooplankton, and particularly small copepods such as Orthona and Onkia and Microcetella, are the gatekeepers of particle flux. These animals are often reported to be associated with sinking particles, and a result of this, we developed a new hypothesis, that particle fragmentation occurs as particle-associated copepods vigorously intercept and search sinking particles for attached protists to eat. And as we've shown in our earlier work on microbial gardening, consuming particle-associated flagellates and ciliates can provide the animals with a nutritionally superior diet to eating bulk detritus. Moreover, fragmenting particles as they search for protists has the additional benefit of retaining particles locally by stopping them sinking, and by making them smaller, this also stimulates the gardening pathway and hence the production of yet more particle-associated protists that can be consumed. And so maybe this certain search and destroy feeding mode has actually multiple benefits. So to test this hypothesis, we used Medusa, an intermediate complexity MPZD type ecosystem model. I don't have the time to describe Medusa in great detail here, but if you're interested, please do search out the ASLO 2013 paper for a full description of the model. But nevertheless, it's important to mention the two main changes that we did make. The first of which was to include an explicit representation of large, fast-sinking detritus, and we did this by incorporating two large particle groups that sink at 35 and 115 meters per day. And the second major change that we made was to include a third group of zooplankton, the particle-associated copepods. We tried our best to really faithfully represent the ecology and physiology of these animals in that they have a low maximum feeding rate, a low feeding threshold, meaning that they're able to feed even when food concentrations are low. They can effectively and efficiently locate sinking particles, and they've got low rates of respiration and mortality. And finally, we implemented the search and destroy feeding by these animals by only allowing them to consume 20% of the encountered large detritus, with the remainder being fragmented into small and slow sinking particles that could then be colonized by bacteria and protists. Whereas we allowed them to eat 40% of the encountered small and slow sinking detritus, reflecting the increased prevalence of protists on these particles. We ran the model using simple 1D physics for the Porcupine Abyssal Plain site at which the Gearing et al. Study was conducted. And the upper two contour plots show that it produces what we think are realistic estimates of the seasonal cycles of particle-associated copepod biomass on the left and particle flux throughout the year and down through the water column on the right-hand side. And on the POC flux plot on the right-hand side, you can see clearly that the simulated flux and its attenuation varies considerably over the year, as you would expect. The bottom plot focuses in on the key period of export during which the Gearing et al. Study was conducted, and shows POC flux now on the x-axis plotted against water depth. And what I hope you can immediately see is that including the particle-associated copepods, shown with a green line, is that we can realistically reproduce the flux data determined from neutrally point sediment traps, shown as the black points, and the resulting Martin curve estimate, shown as the grey dashed line. By contrast, the red line, which shows when we removed the particle-associated copepods from the model, here flux attenuation by microbial respiration alone really isn't sufficient to attenuate the sinking flux as the observations suggest. And so, to return to the question posed at the very outset of this talk, yes, particle fragmentation by copepods can explain the observed flux attenuation in the mesopelagic. The particle-associated copepods in our model were estimated to account for approximately 80% of the annual flux attenuation of large detritus at the Porcupine Abyssal Plain sampling site. And we therefore suggest that allowing models to attenuate flux via a mechanistic representation of the ecology and physiology of particle-associated copepods may allow models to more realistically respond to future environmental change. And in turn, doing so should help improve future projections of ocean carbon sequestration via the biological carbon pump. Improving our ability to parameterize particle-associated copepods does, however, require new and detailed understanding of the physiology of these animals, such as their ingestion rates and food preferences, their capacity to tolerate food deprivation, and life history parameters such as longevity and mortality. And that's me done. Many thanks for listening. I hope it was interesting, and I very much look forward to speaking with some of you during our interactive session later in the week. Thanks a lot. Bye-bye.